Welcome to the second session on executive functions, and I would like to note in the beginning that the lecture of last week is available on YouTube under that link. This is just a short version of this long link, and it's just in several parts because it has to be split down to upload. And the ones of this lecture I will upload as well, but again, it may take up to a week until uploading is finished. And one person, I think she's not here, asked last time for some papers which in more detail specify the relation between MRI studies or imaging studies and patient studies, and these would be two good resources to start reading in more detail about that. Okay, I would like to start with a summary of last week because we covered quite a bit of material. So, we started with cognitive theories on executive functions, and just as a brief reminder, we had as a kind of working definition, we said that executive functions are mental processes for the management, that means regulation and control, of other cognitive processes, in particular in relation to complex actions. We said that there are some key functions which are executive attention. That means we need specific attention to select what we need to process to achieve our goals. Task switching is a typical executive function. That means moving our attention between tasks. Inhibition of irrelevant information which may distract us from our goal. The planning and sequencing of activities, that we do them in the right order, and task monitoring, that means making sure that everything works correctly. Then we had this example of preparing complex meal like a potato gratin, where we said we have several steps which we have to do in a certain order. We may do two different things in parallel, have a multitasking situation, and then we have executive functions which make sure that such complex tasks can be performed by us. As a theoretical background, we briefly spoke about the working memory model according to Baddeley and Hitch, where we have like short-term memories and a central executive system which controls the processing. Another theory we spoke about in a bit more detail is the theory of the supervisory attentional system, and this was the key diagram of that system. The idea was that every action, small actions and complex actions, can be represented by schemas, which are like scripts to work on an action, and that these schemas can be triggered by external sensory stimuli. I may see the dirty dishes in my kitchen, and that triggers the schema, okay, clean the dishes. And a complex schema may consist of several sub-schemas. And we said that one of the major components to organize complex actions is the SAS, which then can, via these, how they call it, vertical threads, may influence the activation level of the schemas and therefore reweight them so that we may be able to, or that we are able to perform schemas or actions which are actually not the highest triggered by the environment, so that we are not solely driven by external stimuli, but have some internal control as well. We said that a problem of that model is that what is the SAS? Where does the SAS know what action I would like to perform next? And so that we have a slight homunculus problem here. And one alternative approach was that we said that, proposed by Jordan Grafman, we said, okay, we can have model of action control solely by memory representations, and he proposed these managerial knowledge units, MKUs, which just somewhat like schemas describe actions on different levels of hierarchies. So there are very specific ones for doing exactly one specified action step by step, and very abstract, context-free semantic units. And according to John Grafman, he thinks that such a high-level control structure as the SAS can basically be described by just a very abstract, such a MKU, and these MKUs are learned during childhood so that we can solve the problem of the homunculus, or what is the highest level of control here in this model. In addition, we spoke about the neural correlates of executive functions, and in particular the role of the lateral prefrontal cortex. We had this diagram where we said executive functions are well localized in the lateral prefrontal cortex because this area not only receives information from all sensory areas, sensory domains, but as well from subcortical structures about body states, hunger, thirst, tiredness, and our emotional states. So it gets all information necessary to make informed decisions, and then it has the power to influence our action system via premotor and primary motor cortex. One important thing we spoke about is the organization of the lateral prefrontal cortex. Is it one uniform area, or are there subdistinctions? And we had first the process-based theory, which said, okay, we can subdivide the lateral prefrontal cortex into a ventral or lower part, or a dorsal, superior or upper part. And the process-based theory says that the ventrolateral prefrontal cortex is mainly associated with the maintenance of memory information, while the dorsal part is associated mainly with the manipulation of information. This example, if I give you my phone number and you just repeat that in your head, it's maintenance. And if I then tell you, oh, please switch the last two numbers, then it would incorporate an additional component of manipulation because you have to do something with that. And this idea roughly fits the idea of the model of Bentley, where you say you have separate memory systems which are more ventrally organized, and then you have the central executive system, which is more dorsally organized. An alternative system was a content-based theory which specified that ventral areas are memory for objects and dorsal areas are memory or maintenance for spatial information. And in this model, in the ventral area, we have many maintenance as well as manipulation for object, and in dorsal the same, maintenance and manipulation. So there's kind of a revised version of this model saying, okay, we integrate that for logical storage into the executive component, visual or spatial stuff into the executive component, so that we have both here. We said this is mainly derived by animal studies, and meta-analysis with brain imaging studies favored the process-based theory, because when you look at where the activation patterns across many studies distribute, then you see the red dots, maintenance only, is mainly ventral, and studies which use maintenance plus some manipulation component mainly from dorsal activations. So that in humans, the process-based theory is the more likely one. Then we turn to the dysexecutive syndrome or frontal lobe syndrome, because it usually or most often happens after frontal lobe damage. So that means the clinical side of executive function, or in this case dysfunction. And the dysexecutive syndrome basically is characterized by problems with all the core executive functions we spoke about. And clinically, this may be evident as perseveration, distractibility, utilization behavior, all the things that we spoke about. So, perseveration, we said we can test using the Wisconsin card sorting test. It was the test where you have to sort the cards according to different rules. We can test for impairment of distractibility or inhibitory control by using the Stroop task, where you have to name the ink instead of reading the word. Monitoring can be tested by the self-order pointing task, where you have to point to all colors in sequence and the colors are reshuffled after every planning, Tower of Hanoi or Tower of London task. And then we said we have this utilization behavior where people just respond to external stimuli and problems with social behavior as described by the case of Finner's gauge. Okay, so much summary of last week. Do you have any questions regarding that? Okay, so let's turn to some new stuff. And I would like to spend now some more time on cognitive paradigms of tasks which fall into the domain of being classified as tasks which demand executive control or executive functions, because so far we have only seen very few tasks and there are quite a few more tasks, and of course we talk about their function in the anatomic correlates as well. So, when we want to improve our understanding of executive functions, we can do this in different ways. One way would be to have a very close look at the cognitive tasks we are using. So, what we can do is, let's say, a task analysis. So, we take a task, let's say the Stroop task, and we look, okay, what exactly is required when I have to do the task? For instance, I may have to inhibit the tendency to read the word, the information. I may have to refocus my attention on the color. I have to set up my attention system. I may do some general preparation. Okay, this is now a situation I'm experiencing conflict, so I'm generally more attentive in that situation. And then work from that on and say, okay, these type of tasks require these type of detailed processes. So what's involved in that? And then we can come from a different area and say, okay, what about functional brain anatomy? We can say, how are the different mental processes, as we have defined them here, are localized in the brain? Are they localized at the same place, at different places? Is there some coherence? And depending on the overlap and dissociation of processes, we may make inferences about whether they are related to each other or not on a cognitive level. So we can very closely and in an interactive way try to define and better understand executive functions by having a close cognitive perspective and functional neuroanatomy perspective. Okay, let's start with the Stroop. We talked about this before. So Stroop is basically that, or the difficult condition in the Stroop task is if you have to name the ink color of the word and it's not congruent, as in this case, where the word and the color match, but it's incongruent, like in this case. Then we have conflict, which is difficult. And there has been a study which tried to dissociate at least two component processes in this type of task. And the first one is our brain systems related to the detection of the conflict. That means the monitoring of the situation and the signaling, oh, we need additional control here because we have conflict. And the process associated with adjusting our behavior, our preparing or implementing of the executive control. And this is a study by MacDonald and colleagues from 2000, published in Science. And they attributed these two functions to do two different brain areas. One, so this is the top left view on the brain, and one is the left dorsolateral prefrontal cortex. We know that area from last time. And this one is an area called anterior cingulate cortex, or in brief ACC. And this is on the medial surface of the brain, so between the two hemispheres. And it's the blue shaded area in this graph. So it's part of the frontal cortex, but it's not part of the neocortex. It's part of an older cortex. It belongs to the limbic system. And we are interested in the posterior part here. The more anterior part is a more affective domain, and sometimes related this area transition to social cognition. Yes. And this more posterior part is a more cognitive domain. So, what have the authors done to disentangle these two processes? They had a standard Stroop task, where they presented words in different colors, which could be congruent or incongruent. However, what they did in addition was, on every trial, to specify whether the participant is required to read the word or to name the color. And word reading is the easy task, because color doesn't interfere very much with that. Color naming is a difficult task. And then they separated that in time, so that in the MRI you can see the neural responses to your instruction. And so one single try to 25 seconds, half a minute. So these type of designs are extremely boring to do as a participant, because every half a minute you are allowed to do one key press. That's all. And then you do that for like half an hour or an hour in the MRI. So you get the instruction, and then you have 12.5 seconds, and then a word comes out. And then you have 12.5 seconds and the instruction comes out. And you probably have heard about MRI, that the reason for that slow pace is that the neural response is pretty fast, but that the blood response, which is measured by MRI, is pretty sluggish. So if you have an event, you get a peak after like 6 seconds or something. So that's the reason why you need to separate them strongly. Okay, so they did that, and then they looked at two factors basically. One is, how do these two brain areas respond to this difference? In red, word task coming up, word reading, that's the easy task, or color naming, that's a difficult task. And then they looked at, okay, when we present the word, what's the difference between congruent, that's easy, the easy task. So if I have to say, okay, this is written in red and it isn't red, and then we have the incongruent case, this is a hard task. So here we have actual response conflict, and here we have a difference in what we expect. And we know the color task is coming up, then we know, okay, now a task is coming which is difficult, which may potentially lead to an erroneous answer.
Can I ask a question?
Yes.
How do you establish that it's difficult for somebody, because it would differ on their literacy and their language, for instance? So for instance, if you had that in a language you didn't know, it would be completely different, wouldn't it?
Yes, because then if you don't know that this word means red,
Yeah.
then you wouldn't have any interference.
Yeah.
And you know that it's difficult by the behavioral data. So people become slower and make more mistakes.
Right, but do you have a range of where those people are when you start testing them? Because I'm dyslexic, and I wonder if it would be different from that, because I learn words as pictures as opposed to reading them. So in some ways the colours might be easier for me.
Okay.
I'm not sure, but
Yeah.
I'm just wondering.
To be honest, I don't know how dyslexia now is taken into account in these type of studies where reading is important. Usually I would say if they want to make claims about the normal population, which is not affected by
Yeah.
anything, then you will make sure that you will test certain
Yeah.
people, and you have specific studies testing on how it's true in dyslexia.
Yeah.
So, but how it is in dyslexia, I don't know.
Okay.
So, but in normal people, this is a pretty strong.
Yeah.
And there's quite a bit of research going on, and you can show Stroop in different variants for different. It happens basically as soon as you have two interfering informations, and here that it is only one way around, so to say,
Yeah.
is basically because we are so extremely well trained in reading and ignoring color.
Yeah.
Because we have so much experience in reading words of different colors.
Yes. Yeah.
So if you create different paradigms, which are not based on reading, but for instance on pictures, picture painting, and things like that, you find a more balanced Stroop effect in both directions.
Yeah.
So that even to transfer that to this situation, reading the word, the color would interfere with
Yes.
that.
Yeah.
And if you do it very carefully, you can even find slight effects in most normal people, that the color slightly
Yeah.
may disrupt you sometimes.
Yeah.
So, but that's the idea. Yeah, so generally you would assess by the behavioral response that Stroop has been present and interference has been present. Okay, so, sorry, very dry mouth today. These are the data for the lateral prefrontal cortex. And as you can see, lateral prefrontal cortex shows specifically activity when the difficult task is expected. The task which may result in interference, while it only shows moderate activity for the easy task. So this, while when then the actual word comes up in the Stroop task, there's no difference whether the word is congruent or incongruent. So this looks like lateral prefrontal cortex is like implementing executive control in advance. You know the difficult situation is coming up, so activity goes up, and everything is set into place so that you will be able to respond in the correct way. Now, as you may expect, anterior cingulate cortex shows the exact opposite pattern. So it does not respond to the cue, what is coming up, easy or difficult, but it responds to the actual word itself. When it's incongruent, in green, activity is higher than when it is congruent. So it is assumed that the ACC, anterior cingulate cortex, has some role in monitoring for conflict and signaling, oh, I have right now detected some conflict. And this signal then may be used for the next try to adjust behavior, try the lateral prefrontal cortex. So that by taking a cognitive paradigm where you just don't know exactly what is going on from a cognitive side point of view, by an MRI study, you can disentangle processes, sub-processes, and you can assign them to different cortical areas, like in this case. Okay, let's turn to another frequently used task, and that is the go-no-go task. Do you know the task? Okay. The name pretty much gives it away. And sometimes you have to go, and sometimes you have to go. So, an example is, for instance, you just have to do a simple response task, and every time you see a number, press a button. However, don't press the button when the number is a 4, for instance. And so that these tasks work nicely, what is usually done is that there's quite a bit of time pressure put onto the participants, and this can be achieved by giving a very limited time window to respond to the stimulus. So if you're too slow, you get a timeout error. the next stressors. And by that, you can push participants really to do very fast. And then it becomes very hard to say, okay, what's the number? No, it was 4, and to push the button. A closely related paradigm is the stop signal task. And to stay with the same example, it would be respond to a number, but not if you hear a tone, which is given separately. One advantage of the stop signal task is that you can vary it a bit more independent. For instance, you can present a stop signal after the stimulus has been presented. So you can test where is the point of no return. How many milliseconds after the stimulus has been presented, a participant cannot stop anymore. Because for some time, they still can stop. And generally, the more Go trials you have, the harder it becomes to stop or to not go. Which is somewhat natural, because if you do something all the time and only then you have to withhold a response, then it's much harder than you have to do it on every third trial. Okay, a question which is of theoretical importance here is whether in the go-no-go task, whether we are really talking about inhibition of the response. Because that's what the go-no-go task is supposed to assess, that we want to respond, but we have to inhibit that. An alternative interpretation would be, maybe it's early attentional selection in the example. Maybe we can set up our visual system that it just ignores the 4, the number 4, if we're not supposed to respond to 4. And then it would be no response inhibition at all. So Logan did a study in 1983 where he did, let's say, a rather complex stop signal task, and he presented questions like that. Is a pomegranate a fruit? People had to say yes or no, or respond with key press, yes or no. And on occasional trials, he gave a stop signal before participants answered to that. Now, what they did after the end of the session is that they did a memory test for certain keywords, like pomegranate, for instance, which was not expected by the participants. And he was able to show that the words in Go trials, or in trials where they responded properly, and in trials where they had to stop and withhold, memory was identical. And that speaks against early attentional selection, because if something is filtered out already very early in our system, then it's very unlikely that it's transferred to memory to the same level or same depth as items which have been fully processed. So if this would have been true, you would have expected that the words from the stop trials would have been remembered worse than the other words, but this was not the case. So one can be, from this cognitive experiment, be quite sure that it's actually really inhibition of the response. Okay, what happens if we do that task in the fMRI, brain imaging? And here we have a study which used a very simple go-no-go task. Green square, you go. If the square is red, you don't go. And then we look what happens, are the brain areas specifically active during the no-go trials as compared to the go trials. So, and what the authors have found is this area, and this is actually the right hemisphere. You may be aware that for these type of images, there are two conventions how to present them. There's the neuroradiological convention and the radiological convention. So, and I always mix that up. One of them shows it right on left and left on right, and the other one shows it left on left, and which can be quite a pain if you do MRI analysis, because different software packages have different standard values. And if you just have such a brain, it's not that easy to say, oh yeah, I'm sure that this is right. And in most software packages, you just change one parameter, flip left, right, And it's just the other way around. And so what some people do is they attach something to the head of the participants, like a vitamin E capsule, which nicely shows in the MRI, and they always attach it on the right side of the head, so that they can be sure, okay, right is on the right and left is on the left. Yeah, but this sometimes can be a bit tricky. And so in this area, which is the right inferior frontal gyrus, so right lateral prefrontal cortex, we see there is no signal change for go triads when people do something, but there is quite a bit of signal when they don't do something. So this is kind of funny because we get MRI signal when there's actually no overt behavior. And so it's assumed that the right inferior frontal gyrus is associated with inhibition, and in particular inhibition of responses. This lateralization effect is independent of response end. So no matter whether people do the task with the right or left hand, this no-go related activity is always in the right hemisphere. So it's not as simple as that. Okay, in a further study, Konishi investigated in more detail what about the relationship of go-no-go triads, and that again is right hemisphere, and this is right inferior frontal gyrus, and the associated activation in the Wisconsin card sorting test. And in this Wisconsin card sorting test, we said, okay, inhibition is probably a very prevalent executive demand as well, because when I have to switch my set, my rule, how to sort the cards, I have to inhibit the old rule so that I'm able to engage to a new rule. And so when you have to do this shifting, you find an activation which is pretty much comparable to the other activation as well. This is in detail the overlap. So the colored pixel are the ones of the go-no-go task, and the encircled area is of the Wisconsin card sorting test, the set shifting. So we find here a very, very striking overlap of the two areas. So that we can conclude that in the Wisconsin card sorting test, we have the same type of inhibition as in the go-no-go task. However, in the same time, let's go back, they have shown that Wisconsin card sorting test is more than just the go-no-go, because as you can see in the left lateral prefrontal cortex, we have activation in the Wisconsin card sorting test, which is not active in the go-no-go. So Wisconsin card sorting test requires more than just inhibition of the old rule, like searching for a new rule, memory updating, what have I used before, how did I respond before, to instate the new rule, to find out. Okay, we have that. So we've seen that for the go-no-go task and the Wisconsin card sorting test, both are used as executive tasks, for instance, for testing of patients. And that both may involve some form of inhibition. In the term the go-no-go task, it's a prepotent response, which I'm likely to press, but I had to withhold. In the Wisconsin card sorting test, it's a more abstract category how to sort cards, and not directly a finger press, which I have to respond. So the question is, are the cognitive inhibition processes the same or not? And are they related to each other? And the study we've just seen suggests